name is Julianna Porter. I am an undergraduate at UC Davis, and today I will be presenting on genome size variation due to repeat content as an adaptation to local climate. Genome size varies tremendously across the tree of life, and we know that this variation is not correlated with organismal complexity. And to illustrate that, I have just a couple examples, including the Antarctic midge, which has the smallest insect genome size, the California two-spotted octopus, which has a genome size very close in size to the human genome. And then our large genome examples include the axolotl and this Japanese flower that has the largest plant genome size. We also know that genome size is not correlated with gene number, at least in eukaryotes. So it's rather linearly correlated in prokaryotes, but that is simply not the case once we get to eukaryotes. And so this kind of begs the question, if not organismal complexity or gene number, why do we see such diversity in genome size? And I think that by attempting to answer this question, we can uncover a lot about how diversification emerges as a whole across the tree of life. So I want to bring your attention back to this previous figure, specifically down at the bottom where we have flowering plants, because as you can see, there's a tremendous amount of diversity in genome size within this group. And that is especially illustrated by these examples that I have of the smallest flowering plant genome and the largest flowering plant genome. And this variation in genome size has been attributed to the fact that this group has had a whole genome duplication event early on in their history. And whole genome duplication events can be attributed to speciation or diversification. And it may be because of this that this group has had such tremendous success. And so that alone makes this a really interesting group of organisms if you're trying to study genome evolution. But not only do they have tremendous diversity between species, flowering plants also have some notable examples of species that have a significant amount of variation within species. And so the one I have depicted here is for maize. And so we have different maize populations on the bottom with genome size on the y-axis, either as a ratio or an absolute value. And yet, despite this significant variation that we are aware of, research on intraspecific genome size variation is fairly limited. One study that does examine genome size variation within species was conducted by my lab, and they looked at how genome size varied along two parallel altitudinal climbs, one in Mesoamerica and one in South America, in both maize and its wild relative teosinte. And what they found was that genome size was negatively correlated with altitude, and they found that this trend held up even when they controlled for population size. And what this means is that this variation is not simply a product of genetic drift, but rather is being maintained by natural selection. Then they took it further and they created a mechanistic link for how genome size is being selected for. And they found that genome size was negatively correlated with cell production rate, and that cell production rate was negatively correlated with flowering time. And so overall, this created a trend that smaller genome sizes were being selected for at higher altitudes through the selection on earlier flowering time. So this was a really interesting finding that we wanted to build off of and kind of conduct similar analyses on in another species. And for that, we chose Trypsicum dactyloides. And we chose this species not only because they occupy a similar distribution as maize, they're found all throughout the Americas, but also because they are found on the same altitudinal climb as maize, which makes these guys all the better for comparison. And not only that, maize and Trypsicum dactyloides are closely related species, and they have a common whole genome duplication event in their common ancestry, which again just makes these guys super interesting when studying adaptation and genome evolution. And so we ended up sampling from 8 different populations, which are depicted on the graph below. These populations belonged to 2 different subspecies of Trypsicum dactyloides, which is indicated by the shape of the point on the graph. And so those belong to either Hispidium or Mexicanum, which are both found in Mesoamerica. And then we had a total of 42 individuals with an average of about 3 individuals per population. We sampled from an elevation that ranged from 10 meters to 1,750 meters, and that is indicated by the color on the graph. And then lastly, all of our individuals were tetraploid individuals. And so we initially started our analyses by using flow cytometry to estimate genome size. And what we found when examining how genome size correlated with altitude was not much. There appeared to be no significant correlation between elevation and genome size in Trypsicum dactyloides, which is not what we saw in maize. Similar analyses that we conducted looking at genome size and longitude or genome size and latitude also came up with no significant correlations. This was a little disheartening, and we went back and tried to look at more research on the subspecies we had selected. And while research distinguishing these subspecies was rather limited, I did find this paper that looked at the genetic relationships within the Trypsicum genus, and they constructed this phylogeny depicted here. And so the 2 subspecies of this experiment are underlined in red. And so the first thing that jumped out to me was that they are not sister taxa on this phylogeny. But what's even more interesting is the other subspecies of Trypsicum dactyloides that were included in this study are underlined in blue. So this supposed single species is seen across the whole Trypsicum genus and just is not as related as we would have expected from a subspecies. And so going back to our previous data and including the subspecies information, we see a completely different story. So now, rather than no trend, we're seeing 2 opposing trends between elevation and genome size. So one subspecies has a positive correlation between genome size and elevation, and the other subspecies has a negative correlation between genome size and elevation, which matches what we saw in maize. And so this was super exciting, but we are not done yet. And so the first thing we want to do is quantify the repeat abundance to identify what parts of the genome are responsible for the variation that we're seeing. And to do that, we first sequenced all of our individuals, and then I called for single nucleotide polymorphisms and filtered those. And then after that, we had to identify which repeats we want to look at. And so the figures I have depicted below are from the previous study. So they found that transposable elements and 2 different heterochromatic knob repeats were significantly correlated with altitude. And so we'll be looking at those repeats as well as some other repeats that are specific to Trypsicum. And so we already grabbed those sequences and we aligned them to our reference genome using BLAST, and then we filtered those results as well. The last step that we have yet to finish is to actually calculate repeat abundance of each repeat for each individual, and we'll be using this formula here below. After that, we will be looking at population structure. And so to do that, we first estimated genotype likelihoods using a software called EVG. And we use that to generate allele frequencies, which we need for a kinship matrix to establish the relatedness between organisms. And we'll also analyze population structure using a software called entropy. And so we'll combine all of those results into a linear model that expresses genome size as a function of altitude relatedness and repeat abundance. And the reason, again, that we're doing this is to make sure that the variation we're seeing is not simply a product of our population structure or the relatedness between our samples, and is in fact being maintained by selection. And so I wanted to conclude with this a reminder of that really exciting trend that hopefully we'll be uncovering a lot more about, and then connect it back to that previous study that not only found that genome size is being maintained by selection, but that it's connected to an organismal function that, like flowering time, that is so essential to a reproductive success of this plant. And then those results plus this little cartoon here on the right, I think highlight that there is still so much we don't understand about genomes and how they evolve. And that by looking at how genome size is evolving at the population level, we can hopefully gain a better understanding of how genomes are evolving across the tree of life. So lastly, I would like to thank, of course, everyone in my lab, all of our additional collaborators, and all of our funding for making this research possible. Thank you.